Hello, now let's talk about hemostasis. Hemostasis is arrest or stoppage of bleeding. Test for hemostasis is platelet count, bleeding time, clotting time. Stages of hemostasis: first is vasoconstriction, second platelet plug formation, and third is blood coagulation. So, vasoconstriction by vasoconstrictors, platelet plug formation by aggregation of platelets, and blood coagulation, formation of prothrombin activator by intrinsic or extrinsic pathway. Now let's talk about these pathways. So, mechanism of blood intrinsic and extrinsic pathway. So, process in which blood loses its fluidity and becomes jelly-like mass few minutes after it's shed out is called coagulation of blood, right? So now let's next learn about stages of blood clotting, that is formation of prothrombin activator either by extrinsic or intrinsic pathway, then conversion of prothrombin to thrombin, and conversion of fibrinogen to fibrin. So our sole aim for intrinsic or extrinsic pathway is for the formation of prothrombin activator, and the sole aim for prothrombin activator generation is for the conversion of prothrombin to thrombin, right? So now let's talk about intrinsic pathway first. So first, injury to blood vessel and exposure of collagen occurs. Then inactive 12th factor converts into active 12th factor, active. Then this active 12th factor initiate inactive 11th factor to get converted into 11th active factor. This 11th active factor now initiates inactive 9th factor to get converted into 9th active factor. Now to this conversion, calcium 8th factor comes into play and converts 10th inactive factor to 10th active factor. So 10th active factor reacts with platelet and phospholipids and factor 5 and becomes prothrombin activator, right? So we can see that this is a cascade process. Now what do I mean by cascade process? It's like as the water flows from river steps, its intensity and frequency increases. So as when 12th factor is inactive is converted into active, that's slow. Then the frequency is enhancing, right? Okay. So now let's talk about extrinsic pathway. The sole function, you have to remember, kids, the sole function is for conversion of prothrombin to thrombin by formation of prothrombin activator. Please remember this. Now, extrinsic pathway, injury to blood vessels, release prothrombin, prothromboplastin, that is third clotting factor, right? So 8th factor converts into active 8th factor. This was inactive. Now inactive 10th converts into active 10th factor, and 10th factor forms prothrombin activator. That was our sole purpose. Now let's talk about fibrinogen. Fibrinogen converts into fibrin. Then fibrin thread get attached to loose platelet plug. This blocks ruptured part of blood vessel and prevents further blood loss completely. This was the coagulation, that is formation of prothrombin activator by intrinsic or extrinsic pathway. Now let's talk about something which is really opposite to it, that is anticoagulants. So what are anticoagulant? It prevents clotting. Example, salts of citrate, oxalates, EDTA, these are chelating agents. Please remember this. Heparin and dicumarol uses our storage of blood, lab purposes to maintain fluidity of blood. Anticoagulants, there are two types, in vivo and in vitro. In vivo is inside body, heparin and EDTA. In vitro is outside, used outside, right? That is citrate and oxalate. Now let's talk about